Research Summary Group 1. Hello everyone, warm greetings to all present. As we gather here today, we are excited to introduce our research summary part, which aims to bring a new point of view to anatomy and surgery with different usage of the nerve. Our group is consists of Ilkin Mehmetli, me, and Murat Jafarli. And our advisor in this research topic is Professor Nihal Apaydin from the Department of Anatomy. And let's switch to overview of our slides. And today we'll talk about mainly the topic and aim of our research project, research question, hypothesis, outcomes, variables, data collection techniques, and inclusion and exclusion criteria. Today I'll talk about topic and aim, research question, and hypothesis, and my friend Murat will continue to talk about outcomes, variables, which are independent and dependent variables, data collection techniques, and inclusion and exclusion criteria. And let's begin with topic and aim of our study. Our topic is anatomy and quantitation of the lower subscapular nerve and its potential use for the nerve transfer surgeries in upper limb. And our aim is to evaluate the anatomical structure, axonal composition, and clinical feasibility of the lower subscapular nerve as a donor nerve for upper limb nerve transfer surgeries. And let's switch to the research question. Our question is that, can the anatomical structure and quantitative potential of the lower subscapular nerve be used in nerve transfer surgeries to restore function in upper limb injuries? As you can see from this question, we'll mainly focus on to find the anatomical structure of lower subscapular nerve, the feasibility of lower subscapular nerve in upper limb nerve transfer surgeries. We'll use anatomical measurements, some histological measurements, which my friend Murad will talk about this later. And let's switch to the main part, the hypothesis of this research project. Our hypothesis is that the lower subscapular nerve has sufficient anatomical length, regional location, axonal count, and functional properties to serve as an effective donor nerve for upper limb nerve transfer surgeries, improving functional outcomes in upper limb nerve injuries. As you can see from this hypothesis, we'll try to confirm that the lower subscapular nerve is efficient nerve for usage of upper limb nerve transfer surgeries. In order to confirm and find this hypothesis, we'll use anatomical structure and anatomical length measurements and histological techniques like axonal counts and so on. And my friend will continue to talk about our research project too. And thank you for listening to me. We can switch to the Murad's part.
Hi there, I am Murad Jafarli. Thank you, my friend, to introduce the study. First, the aim, questions, and main parts, and I will continue with the secondary parts like outcomes, variables, our data collection techniques, and so on. So let's start with outcomes. As you can see from the slide, you can see primary and secondary outcomes. First of all, the main part of our study, the outcome from our study, we can see the feasibility of the lower subscapular nerve as a donor nerve for nerve transfer surgery. You can think that it is a plastic surgery field, but we introduce our study in anatomical part and also in surgical part, of course. And we want to see the feasibility of this nerve and how can we use this in the surgery field and so on in the future, of course. Then you can see secondary outcomes, innervation potential, recovery ability in surgical transfer area, and surgical ability of donor nerve area. What can we understand from secondary outcomes? You can see the generally potential of this nerve from innervation, and this recovery ability is also the main prospects in the transfer surgeries in a surgical part, and also we see the anatomical part. What donor nerves can be used, for example, the other nerves, and so on. So let's continue with variables. Variables, we write there, our study focuses on cadaver dissection, related results, and their potential usage in nerve transfer surgeries in the future. As you can see, the dependent and independent variables. For example, our dependent variables are functional post-surgical outcomes and post-surgical complications. They are dependent variables because in cadaver dissection, these parts are mainly impressive. So the independent part is anatomy of donor nerve, length, strength, area. In this field, we study in brachial plexus. That's why the area, the length of nerve are more independent than dependent variables, and also axonal density of donor nerve and cadaver related factors: age, sex, weight, ethnicity, muscle mass. These ones are mainly cadaver based problems, so this variables doesn't affect more in our study because we generally look at the anatomy and the potential usage in surgery. Let's continue with next slide. What's our data collection techniques? As you can see, we write and show the pictures of them because of the clearly understanding from the slide. Briefly, we use related sources from PubMed, and we used cadaver dissection and also histomorphotomy in peripheral nerve. As you can see from here, the three parts of our study. Firstly, we get a literature. We expand our literature knowledge about in PubMed. Then we studied in cadaver dissection, so we can understand the nerve transfers very well. So we want to study it in histological part too, because the feasibility is not only anatomy; it also calls the histology part too. That's why we want to work with the histology department in this field. So let's continue with the next slide, our inclusion and exclusion criteria. As you can see, there are mostly more in criteria there. It is mainly about cadavers because our university is rich for cadavers from the other universities, so we are so lucky in this field. So inclusion criteria is about adult cadavers, fixed cadavers, and full body cadavers. These are the general types and features of the cadavers. And so exclusion criteria is the mainly the region or some general aspects of the cadavers. As you can see here, cadavers with the brachial plexus region previously dissected and injured, and also in upper limb we can see there. And one of the another exclusion criteria is about cadavers in which the origin, insertion, and penetration points of nerves or muscles cannot be determined. These are the general criteria, and we will finish our slides in there. So thank you for all to our listening, and have a great day.